Hello everyone, my name is Tolulope and I am excited to attend this Epigenomics SIG meeting, and I am honored to have been selected to receive the Epigenomics SIG Student and Investigator Award for 2022. I'll be making my presentation titled Bisphenol A Associated Epigenetic Changes in Young Adults in Northern Nigeria. I have no conflict of interest to declare. Endocrine disruptors are chemicals that may make hormonal activity, interfering with the functioning of endocrine systems in various living organisms, and it can be found in a variety of household products. A typical example will be bisphenol A, which is used in the manufacture of polycarbonate plastics and the lining of cans. There has been concerns about potential health risk of bisphenol A because it has a strong estrogenic effect and it can compete for the binding of estrogen receptors compared to estradiol and other hormones. BPA mechanism of action is linked to the hypothalamus pituitary gonadal axis because the gonadotropin releasing hormones stimulate the release of other important hormones. Studies suggest that environmental exposure to BPA during critical windows of development is associated with genetic changes and reproductive abnormalities, leading to adverse health outcomes. BPA is a known inducer of estrogen receptors and steroid receptors, and it can modulate the transcription of downstream genes, hence leading to the altered expression and methylation of other genes. And alterations in the methylation of these genes have been associated with several human cancers. Even though BPA exposure remains a global concern, BPA exposure assessment studies in Africa are nascent and are poorly characterized. It has been reported that BPA exposure is not ubiquitous in the general US population. With the unregulated use of BPA in consumer goods and the increased utilization of BPA-containing products, there is a need for the biomonitoring of BPA within Africa. And to assess the extent of research that had been done on BPA exposure in Africa, our team identified, collated, and summarized the levels of BPA that have been reported across Africa by performing a systematic search of indexing databases to identify articles, and we extracted the necessary data from the selected articles. Of the 42 publications we retrieved, only 20% of the 42 publications analyzed BPA in human biological fluids, and this is worrisome. Young adults and children are particularly at the receiving end of increased urbanization and use because they make use of plastic packaging, canned foods, and they're exposed to thermal receipts and other consumer and personal care products, which are all major exposure routes to BPA. Based on this, the focus and the goal of our research was to evaluate the different exposure sources to BPA in the population and to investigate associations between BPA exposure, reproductive hormones, and DNA methylation changes. This population was selected for this pilot study because young adults are particularly at the receiving end of increased urbanization because they make most use of plastic packaging, canned foods, thermal receipts, and other consumer and personal care products, and it is also an important time point in their lives for their reproductive health, with the hope that the findings will lead us to expand our research into other populations. Healthy Covenant University students within the ages of 18 to 24 years were recruited, and the demographic and anthropometric data, the eating habit and frequency of food consumption, was gotten from the administered questionnaire. Whole blood was collected and DNA was isolated from this whole blood, and urine was also collected in BPA-free bottles. Hormonal and BPA assays were done using ELISA, and several hormones, reproductive hormones, were assayed for. The DNA that was isolated was then bisulfite converted and biosequenced to detect the DNA methylation changes at OX-A10, PPAR alpha, and estrogen receptor sites. Our results showed that BPA was detectable in all the urine samples that we assayed for, but the range in our study was lower than what has been reported in other continents. Few studies have been done on BPA levels in biological fluids in Africa, so this cannot be a true picture of BPA levels in Africa since studies are limited and there is a dearth of information in the biomonitoring of BPA. Only a few studies on BPA levels in biological fluids have been done in Africa. In measuring the strength of the relationship between BPA concentration, hormones, and DNA methylation changes in males, we saw a strong negative correlation between BPA and peroxisome proliferator activated receptor alpha, which is PPAR alpha at CpG site 1. PPAR alpha is a steroid hormone receptor and it is inhibited by estrogen, and it acts by regulating the expression of several genes that are vital to lipid and lipoprotein metabolism. BPA has been reported to be a known inducer of PPAR alpha expression, and this is because BPA can bind to PPAR alpha and modulate the transcription of downstream genes, leading to altered expression of genes that are related to metabolism of sex steroids, and this can lead to the impairment of the normal physiological functioning of sex steroids. In measuring the strength of the relationship between BPA concentration, hormones, and DNA methylation in females, we saw that BPA was negatively correlated with testosterone and OX8N at CpG site 1, and also with estrogen receptor at CpG site 1, while BPA was negatively correlated with luteinizing hormone. Our research showed that BPA exposure was associated with hypomethylation of OX8N and estrogen receptors at CpG site 1. The homeobox gene, which is OX8N, controls uterine organogenesis, and its expression is affected by BPA exposure, and testosterone is a known regulator of OX8N, so it was not surprising to see a strong negative correlation of both testosterone and OX8N. Alterations in OX8N methylation has been associated with several human cancers, and research has shown that BPA is capable of hypomethylation of the DNA in females, which we saw in our study. Most experimental studies show that exposure to BPA and its analogs can cause decreased testosterone levels due to the impaired levels of luteinizing hormone. Estrogen receptors help in suppressing gonadotropin releasing hormone secretion because they play a role in regulating reproductive behavior, and this can possibly be linked to the hypothalamus pituitary gonadal axis because the release of gonadotropin releasing hormones stimulates the release of follicle stimulating hormones and luteinizing hormones, and both can act on the testes and the ovaries to release testosterone, progesterone, and estrogen. And most human studies have shown an association between the presence of BPA in biological samples and their effects on reproductive hormone outcomes. Multiple linear regression analysis was also used to examine associations between urinary BPA levels and reproductive hormones, adjusting for sex and BMI. We saw that higher levels of BPA concentrations was associated with increased luteinizing hormones and progesterone concentrations, all else being equal. We also saw sex-specific differences in the adjusted models. Females on an average would have about 2.1137 units increase in luteinizing hormones levels than males, accounting for BPA concentration, and this effect was significant at 0.05 level. We know fully well that reproductive hormones have a large variation within a menstrual cycle, and we took this into consideration by restricting the menstrual phases to the follicular phase while recruiting females. After adjusting for sex, we conclude that BPA exposure will be an independent risk factor to increase progesterone and luteinizing hormonal levels in females. Other studies have also associated BPA exposure with adverse reproductive endpoints. Together, this data raises concerns about the potentially deleterious impact of BPA on human reproductive health. In examining the associations between urinary BPA level and DNA methylation changes, adjusting for sex and BMI, we saw that a 1% decrease in BPA concentration was associated with decreases in PPAR alpha at CpG site 1, accounting for sex and BMI. Differences in BMI significantly decreased PPAR alpha CpG site 1 methylation changes by 0.767 units, accounting for sex and BMI. After adjusting for sex and BMI, we conclude that BPA exposure may be an independent risk factor that leads to the hypomethylation of PPAR alpha at CpG site 1 in females. In conclusion, in the estimation of BPA levels in the cohort, we saw that BPA was detectable in the selected population, and the BPA levels in this cohort was lower than the values that were obtainable from other continents. Although this might not be a true picture of what BPA levels might be in Nigeria or across Africa, validation using larger sample size will be required. In estimation of associations between BPA exposure, hormonal and DNA methylation changes, we saw significant correlation between BPA exposure, hormonal and DNA methylation changes, and we also saw significant associations in sex-specific differences in all the adjusted models. There remains a dearth of information on human exposure to BPA and the developmental programming consequences in Africa. The extent of exposure assessment for BPA across Africa, possible sources of exposure, and the range of exposure levels should be further investigated. Due to increased urbanization and the use of plastic packaging and other routes of BPA, it is expected or it is projected that there will be a doubling in the usage of BPA in Africa, and so there needs to be policies regulating the use of BPA in Africa. I would say that an awareness must be created in Africa so that individuals can make personal choices to reduce exposure to BPA. Choices like reducing the use of canned or plastic foods and eating fresh or frozen fruits, or when possible opting for glass, porcelain or stainless steel containers, particularly for hot foods or liquids, and also using BPA-free toys and bottles for children. Overall, BPA research in Africa is still nascent, and there is need for more studies, especially biomonitoring studies, to determine the daily intakes of BPA in Africa and the toxicological impact of such exposures. This study would increase understanding of the impact of BPA on health outcomes and also determine country or population-specific policies on the use of BPA across Africa. I would like to acknowledge the University of Michigan African Studies Center, Covenant University, the Digoot Spy Lab, UMAP's Fall 2021 cohort, and the Rotimi Lab, where all of this work had been done in one way or another. Thank you so much for listening.